Hello, everyone. I'm very pleased to be here today and have the opportunity to present Anemocyte to you, even if unfortunately just on a virtual platform. I hope soon we will have a chance to really see each other face to face in person very soon. Anyway, we thought that the best way to introduce Anemocyte to you was to actually stress what has been recognized as a very unique approach. And so to be a BMO rather than a CDMO, and I will really explain that in a few seconds here shortly, but even more important is to present you the foundation of what a BMO is. That is our Innovation and Development Department. Before entering into the details of the BMO concept, it was very important to give also an heads up on who Anemocyte is and the family we are part of. So Anemocyte is part of the 9 Trees group, is an entity in 9 Trees under which we found several companies, and probably the most representative one is FIS, Fabbrica Italiana Sintetici. This is also the oldest member of the group, has been active since 1960, and the core activities of the group is really to work mainly in active and intermediate ingredients for the global pharmaceutical industry, mainly focusing on products that are already commercially available. And we do that through our main company. This is FIS, as I mentioned, but also with our other players that belongs to the group that are placed all over the world. Even if the group, as I said, has been active now for more than 60 years, it's an extremely active group, up to the point that in the past 2 years, the group have opened 2 new entities, Fulton and Brenta, both entities here in Italy, but we also opened an office in Tokyo. So now there is a physical presence of the group in the 3 main markets in which we are active, meaning North America, Europe, and Japan. Anemocyte is one of the most recent member of the family, and is the only entity that is totally focused on cell and gene therapy. But in any case, it's very important to explain that even if Anemocyte brings on the table more than 15 years of experience of cGMP manufacturing, the 9 Trees group as a whole can provide an additional support to what we can offer. And that will be done, as I said, in a global approach with more than 1,800 colleagues worldwide present. Now, let's really focus on Anemocyte itself. So Anemocyte, as I mentioned, presents itself as a biotech manufacturing organization. So and our focus is totally the cell and gene therapy aspects. What it means is that we proactively look into solution, even if there is not a contract in place. So if you want to compare the BMO versus the CMO, this is really the key elements. This allowed us to have a more educated discussion with you when you come with us as a customer. So we do offer contract manufacturing services, but with a different flavor behind them. And the slide here really represents one poster that was presented last year by our innovation and development team. And you see, 2 keywords are really manufacturability and sustainability. For us, those are really 2 important elements to keep in mind when we think about the providing cell and gene therapy. To enter in more details, when we talk about manufacturability, what we mean is everything that is around the cell therapy product itself. So the key elements of quality, identity, purity, and safety, but also the potency. So your product has to do something. And this is pretty straightforward concept, of course. On top of that, we have to consider the sustainability. So not only the possibility to manufacture the product, but also to make it in a sustainable way. So that means to look at the cost of goods, and the reimbursement, which is the other side from an economical perspective, but also to take care of all the regulatory requirements. And since we are talking about cell therapy, logistics is also another key issue. So we look at these 2 elements, manufacturability and sustainability, in what are the core area of Anemocyte, which is the cell therapy on one side, autologous or allogenic, the non-viral modification of cells. We are strong believer of non-viral, as you will see here shortly, some elements around that as well. And we have also a unit that is totally focused on plasmid manufacturing. Now, let me give you some example, and to give you, the proper starting point. We decided to use the CAR-T as a way to provide you some highlight of the activity that Anemocyte is performing in its innovation and development department. So the slide here really represents in a nutshell the way to manufacture these cells. So you start from blood withdrawal, you modify the cells, you expand them, and you send them back to the patient. Or if it's an autologous product, you freeze, and then again, patient is the place where the cells will go. If we look first at the expansion aspect, there are several platforms that can be used, and it's very important to really have a pulse on how of the pros and cons of the different platform that can be used, because every platform has its own pro and cons, of course. To give you a little bit of an idea on area where we are spending time in the past 12 months, on one end, we have tried to automate as much as possible the G-Rex. G-Rex is a very interesting tool for T cells expansion, and we decided to work with the Lovo, which is a technology that can allow to move fluids and use that as a way to automate a process that otherwise would be very manual. I will not spend too much time just for this to respect the time of this talk on this aspect, but I would like actually to focus more on the second side, that is the media aspect. So media is also an interesting aspect that needs to be properly investigated and evaluated when we talk about cell therapies. So choosing the right media is pretty key. To give you an example, historically we've been using a product that is xeno-free and serum-free with very good success. But as a BMO, we have always found this important question mark, the fact that even if the product is xeno-free and serum-free, it does contain protein of animal origin, and to be more precise, human origin. Every time we try to investigate something that was totally animal-free, we never found a tool that could allow the success or the same results that we were observing with the media we were using. Most recently, though, we opened a discussion with Fujifilm, and they suggest to evaluate their media, the PrimeXV. So what you will see here is a series of experiments we did with the G-Rex platform, the 24-well plate. We followed exactly the extraction recommended by the provider. And the goal of the study I will show here is really comparability between medium A and the PrimeXV. The experiment lasts for almost 2 weeks. The first 3 days are always the activation, and then we monitor on different time points, cell count, and the cells itself. So what you will see here, that was very important, is that first of all, both media can sustain activation of the cells. CD25 is a more later marker, so they're still both of them quite active. CD69 is more an early one, so it is already going down after 3 days, but all expected. What was very astonishing for us, though, is the fact that the PrimeXV not only is completely animal-free, but seems to be able also to sustain a much higher yield of cells at the end. So what is key here is the fact that the cells we are looking at are just T cells, so no modified one. And we need to repeat, of course, this experiment also with the CAR-T cells and see if the results are the same. But what I would like to already pointing you is that according to what is the needs of your product, based on the results we obtain, we could either go in a direction where we can get more cells at the end of the process, and you see on the right side we can get 3 times at least the number of cells we can obtain otherwise, or we can still aim for the same number of cells, but with significant saving on some of the expensive reagents that will enter into place. So what's next, of course, will be to further characterize the cells, evaluate scale-up, and evaluate the CAR-T process, but it's already an interesting starting point. The second area I would like to explore with you is more the gene editing. As you know, the most established way to modify cells is viral vector. But when you talk about viral vector, there are several steps to get into the viral vector itself. So first of all, you need to produce the plasmid. And interestingly, already in 2016, it was highlighted the fact that plasmid will be a bottleneck of the industry. And actually, with more and more requests of viral vector, is becoming more and more an area of concern. we have identified this in an area of investment for us. So in 2019, we made a very big investment in the manufacturing area of plasmid. And if you want to know more about this capacity, we will be pleased to follow up offline. But another segment is really the production of the viral vector. And again, also in this case, in 2017, when the first CAR-T therapy were presented and successfully getting commercial rights, it was already seen as a potential additional bottleneck. In this slide, I summarize all the different red flags that were raised on viral vector in the past few years. I have to say that all of them have been properly addressed, but two I will say: the viral vector manufacturing that I mentioned here, just mentioned before, and the cargo size. So sometimes the size of the gene that you want to transfer is a bottleneck, or is not possible to use such a size when you use viral vector. So there are a few alternatives that you see here, and as an Anemocyte, we decided to invest in the electroporation one. So we did that, opening a collaboration with Maxcyte. We are the center of excellence for the technology, and of course we are aware that by transferring, just by electroporation, the gene of interest will not integrate in the cells. So what is important is also being able to integrate the cells, and to do that, we are using transposon. The Maxcyte technology has been proved to be very successful in integrating in cells different tools, RNA, plasmid, and transposons, as I mentioned. And here shortly, I will just highlight a couple of results we obtained by electroporating PBMCs and then expand them in G-Rex. This is just a proof of concept, so I cannot share data provided by third-party clients. But in this case, we did use a transposon-based technology with a GFP marker. So it was very easy to follow the cells. So on one end, the transfection efficiency was very high. As you can see here, after 3 days, we could see a high signal of GFP. So we know that the electroporation did work well. In terms of viability, it was another important parameter. As expected, we have seen a little bit of drop in viability at day 3, but nothing really dramatic. As you can see at day 7, it goes back to normal. So we're quite pleased about the technology, and we think it can really open several alternatives in the CAR-T space. So in conclusion, I think for me it was very important to stress that Anemocyte is really looking 360 degrees in the manufacturing perspective, or to better say, really vein to vein, by looking at aspects like manufacturability and sustainability. Since I'm getting into the conclusion, I think it's also important to stress the fact that manufacturability and sustainability can be also expressed in cost, unfortunately. So you can not only look at cost of manufacturing when you evaluate the cost of a therapy, but really there are several aspects, the quality, licensing, supply chain, etc., that enter into the play. So you really have to look into globally in your therapy. Anemocyte is really proactively looking into that, and we are generating more and more data around way to optimize the manufacturing procedure. For us, what is very important is really together to look at the sweet spot of your product and make sure that is something that we can implement and start to utilize. Our core business is, once again, cell therapy, non-viral modification, and plasmid. And we do that in the manufacturing side that we have outside Milan. As you can see here, there are 2 separate facilities, one GMP for cell therapy and non-viral modification, one for plasmid. And of course, we have different labs for analytical purpose and the innovation and development. So with this, I conclude. And of course, if there is any question, I'm happy to address them either now on the chat or offline later on. Thank you for your attention.